Welcome to the Onion IQ podcast. This episode is about management of bacterial diseases in organic onions. My name is Alice Formiga of eOrganic, based at Oregon State University, and today I'm talking with Dr. Bhabesh Dutta of the University of Georgia and Dr. Thomas Isakit of Texas A&M University about a research project that they're working on about organic onion production and specifically about soil-borne onion bacterial diseases. The project is called Onion IQ, Improving Quality, Resilience, Soil Health, and Food Safety in Organic Onion in the southeastern and southwestern U.S. First, Dr. Dutta will give a short presentation to introduce the project, and then I'll be asking him and Dr. Isakit some questions about what they've learned so far and some of the different ways to manage soil-borne bacterial diseases in organic onion production in Georgia and Texas.
Thank you, Dr. Farmiga. I am Bhabesh Dutta. I'm a project director of this Onion IQ project funded by USDA NIFA ORAEI in 2023. This is a 4-year project. And I am a professor, an extension vegetable pathologist at the University of Georgia, Tifton campus. I primarily work on onion bacterial diseases and other onion fungal or oomycete diseases. But my program also spans to other vegetable crops, including brassicas, cucurbits, and solanaceous. But for this podcast and for this project, I'll be focusing my attention towards organic onion production and the challenges the organic onion producers face, especially in southern United States. So there are a plethora of onion diseases there, and among those onion diseases, the soil-borne diseases, particularly the bacterial diseases, are the hardest to control or manage. So some of the important examples of soil-borne bacterial diseases and the symptoms are listed in this figure, shown in this figure. You can see on your left is called sour skin of onion. This particular bacterial disease is caused by a Burkholderia species complex, particularly a gram-negative bacterium, Burkholderia cepacia. It leads to pre-harvest and mostly post-harvest losses. The another bacterial disease which we see here in southern United States, particularly in Texas and Georgia, is onion bulb decay or onion slippery skin. Onion bulb decay, you can see, as the name suggests, it can cause complete decay of onion bulbs. The onion may appear healthy from outside, but as soon as you cut open on your kitchen table, you will see the onion rot inside. This particular disease is caused by Enterobacter cloacae, another soil-borne bacterial pathogen. The last one which I mentioned is called slippery skin. It is also caused by one of the species of Burkholderia. It's called Burkholderia gladioli pathovar allelicola. This particular pathogen causes slippery. The outer skin of the onions becomes slippery, and as you try to cut open using a knife, your knife will slip. These bacterial pathogens are notorious and in their survival. They can survive for a long period in the soil. There are very limited management practices available to get rid of this pathogen or eradicate these pathogens from the soil. We cannot control these pathogens, but we can manage them. And these pathogens are notorious, not both conventional and organic onion production, but the management in organic production system is much more challenging compared to conventional production system. So through this project, this is a highly integrated project. We are utilizing different cultural practices. We are utilizing different nitrogen fertilization program, and also integrating the positivities of both the objectives to come up with the integrated objective where we will look at the cultural practices and optimize nitrogen fertilization to reduce not only bacterial pathogens population in the soil, but also enhance the soil health, soil productivity, quality and productivity of the onions. So in objective 1, we are evaluating cultural practices like biofumigation and soil sterilization that would reduce the pathogens, weeds, and also sustain the soil beneficial macroflora and predators and natural enemies. So the one aspect, the one approach which we are taking in objective one is to fine tune a detection assay that can differentiate saprophytic Burkholderia from the pathogenic Burkholderia in the soil. So major problem in differentiating Burkholderia in the soil is we did not have any tool to differentiate them. Through this project, we are utilizing genomics and also molecular tools to find regions in Burkholderia that are specifically associated with the onion-associated Burkholderia compared to saprophytic Burkholderia. And utilizing those assays, we could actually quantify how these cultural practices impacted the bacterial populations in the soil. In objective two is a little different. Here we are trying to see what uh, what nitrogen fertilization regimes we should use in the soil that will not only enhance the soil health but also reduce the harmful bacterial populations. How these poultry litter influence the survival of salmonella or food safety. Are our onions at risk when we use this pasteurized or semi-pasteurized poultry litter? And objective three is based on the positive outcomes of objective one and two. We would choose the fine-tuned uh, expected outcomes, put it together, and then see if in integrated trials if these objectives would not only improve the system but also improve the soil health and improve the yield and productivity of organic onions. As with any NIFA project, extension economics are integrated within each objective, and we hope by the end of the project we'll provide a necessary toolkit for our organic onion growers that will help them to reduce the losses because of these soil-borne bacterial pathogens and with improved yield and quality. So all these objectives, while we are drafting this proposal a year and a half before we started talking to the organic onion growers during different extension meetings, stakeholder advisory panel meetings, or also regional meetings. And they were pivotal in identifying our critical needs. And they are the one who identified these points that we need a cultural practices. We are using cultural practice, but we are not sure if we are reducing or improving soil health. We are also not sure if the population of harmful bacteria in the soil are decreasing or increasing over a period of time using these cultural practices. And they're also not sure about the organic fertility program for nitrogen, particularly for nitrogen, which you're using in the organic soil. They're not sure what is the nitrogen release efficiency and so forth, how it is impacting the overall soil health. They also needed some input about how better they could produce onions so that there'll be less risk of food safety pathogens. Overall, in this project, we are integrating host, environment, and pathogen based information to come up with a comprehensive knowledge that would help our organic growers in growing onions in this conducive disease environment.
Thank you very much. That was a really helpful introduction. Just to backtrack a little bit, can you tell us how much of a problem these bacterial diseases are in Georgia and Texas, and whether they affect growers in other regions of the country as well?
Very good question. So in 2019, there was a project funded by Specialty Crop Research Initiative but to Washington State University, Dr. Lindsay Dutoit. And through this project, we surveyed close to 12 different onion growing states in the United States, and we ranked the most prevalent bacterial pathogens in those states. Apart from Pentoya species, we found Burkholderia to be most prevalent and problematic pathogen across the United States.
Hmm.
Now, in terms of numbers, in Georgia itself, we get pre and post harvest losses close to 4 to 8% strictly due to bacterial diseases. And when we did some of the surveys where we found what are the major causal agents of those post harvest losses, enterobacters, Burkholderias were the prominent ones. And when you talk to Dr. Isaac Kidd, he will also tell you about the problem. The similar pathogens are also problematic in Texas.
Dr. Isaac, do you want to tell us about what's going on in Texas?
Well, we do have bacterial soft rot bulk problems in Texas as well. Um, I've when I've been working in Texas in the first 5 years of my employment, located in the lower Rio Grande Valley, where most of the onion production takes place, the bacterial diseases of onions were not the most prevalent. I see it from time to time, but the growers did have other pressures, pink root, fungal disease, as well as various foliar fungal diseases. But it's pretty important from the standpoint of, well, here's the finished product, and the bulbs are rotten. So it's from time to time it can be a big problem.
So, if a grower has this disease and the onions look fine on these diseases and the onions look fine on the outside, is cutting them open the only way that you can tell that you have these diseases, or is there any other way to tell? I know that you're hoping to create tools for rapid detection.
Well, I'll take this question. So the different
Okay.
uh, stages of disease development in onions. If some of these soft rotters rot the onions, if some of the soft rotters produce aggressive symptoms, you will see complete rotting of onions from outside. In
Mhm.
some cases, the pathogen population is lower, and it can, during the time of storage or during the time of harvest, you not be able to see the symptoms because the population in the onions have not reached to the level, the critical level. In those cases, probably you will see some symptoms at the neck. You will see the softer necks. So when you buy onions, you look at the neck. If the neck is soft or the shoulder is soft, more likely you will have some rot inside. But if the neck is hard, we have seen harder necks, completely nice, well-rounded onions, but after a period of storage, they are disease symptomatic from inside. So depending on the stages of diseases, if you are lucky, you will be able to sort out those diseased or symptomatic onions, or you can have a feel. But those are internally placed or internally localized. It will be very hard. There was a project funded in 2012 through a CRI to one of our engineers here. They tried to find a optical sensing of the internal symptoms, and it was hit and miss.
Mhm.
So we do not have a foolproof, non-destructive way of finding it. So what the growers do, what I have seen, so when they harvest and they keep in storage in a bin, they choose random 20 to 25 samples, cut open and see what is the overall health of the particular bin. It will give you some idea what they are dealing with.
Okay, yeah, that makes sense. Okay, so is there anything interesting that you've discovered in your recent trials so far?
Sure, sure. So as I mentioned, one of the important, I would say, bottleneck in doing these cultural practices, that we are not able to see, or we are not able to measure the impact of the cultural practices in the Burkholderia population. The reason being, in the soil environment, we have saprophytic Burkholderia as well as Burkholderia that are associated with onions. So when you do those culture practices, we are not sure which bacterial population we are actually impacting. So through this project, we have developed a tool based on it's a PCR based or polymerase chain reaction based, where we found there are certain markers in onion associated Burkholderia, and using those markers, we developed assay, both conventional PCR based and also quantitative PCR based assays, to quantify the impact of those culture practices on onion associated Burkholderia itself. So what you're seeing over here on the left, we have onion Burkholderia specific primers. It was able to detect the onion pathogens, and we also have a Burkholderia TTG specific primers that will only detect those pathogens which are associated with onions. They are detected through this assay, where other Burkholderia species which are present in soil as a saprophytic Burkholderia, they were not detected. And we were able to develop a qPCR assay to quantify how much of each how much population of each species are actually prevalent in the soil. And using this assay, now we are quantifying the impact of cultural practices like soil solarization, soil biofumigation, to see to pinpoint the actual impact of these practices to onion-associated Burkholderia.
Dr. Isaac, do you want to talk about anything interesting that you've discovered in your recent experiment?
So in our trials in Texas, there's the locations are split. The uh, solarization trial is done somewhat locally around College Station. The other trial is the biofumigation trial is done in Overton area. And both the soils, they're different soil types, but both fields were not really used for onion production. One has been out of vegetables for years and years. So it's been a challenge seeing effects of the treatments on the bacteria. And of course, the bacteria are introduced on rotted, infected bulbs to the field, and we measure it. We've so far it seems that the bacteria just kind of disappear. They're not persistent as is. But one of the surprising things we saw with the solarization experiment was the appearance of a fungal disease known as southern blight in most of the treatments. And we're figuring, okay, the literature says that southern blight is controlled by soil solarization. We think it might have come in just from the outside of the out of the treated area, but that's something else that could be looked at. It causes rotting of onions as well, and we're interested in pursuing that. And that was the advantage of being a part of this particular experiment, to focus on the bacterial diseases of onions, but at the same time, we get to observe any other kind of diseases that are infecting onions and see how an organic production system would affect those diseases and how we could possibly control them.
So that brings us to our next question, which is how do you approach the management of these diseases in organic productions? And are there any techniques or methods that could already be recommended? You mentioned how challenging it is.
The cultural practices, like I mentioned, biofumigation, soil solution, some of the organic onion growers are actually doing it, but they do not know how much they're getting benefited. Hopefully, through this project, we'll understand empirically that how these practices impact Burkholderia or other soil-borne pathogens. But in terms of actual management, I would say the major management practice uh, happens how you harvest onions. We have seen, particularly with Burkholderia, that how we dig onions, how we cure onions in the field, how we store onions, and how we clip onions has a big impact on the quality of onions. For example, the onions have to be matured in sense the when the 70% of the onion bulbs in the field, the leaf fall down, that's the indicator of the onion maturity. So what happens during that stage, the neck of the onion get dried, and because of dried neck, the onion plant cannot sustain the leaves to be upright, so they follow. So that's also indicator of a physiological maturity of onions. So 70% of the onion should be physiologically mature, and you will see as indicated by falling of those leaves. When the onions are lost, when the leaves are lost, then the onions need to be undercut. We have seen that if you do undercutting through chain digger, you will uproot the onions, the roots faster, and the remaining moisture which is present in the onion neck get dried and get removed pretty quickly. So uprooting of the onions from onion roots from the soil through chain digger is also a very important step. The next step is to field curing. You have to cure the onions in the field. Curing means you have to leave the onions in the field under sunlight so that the onion neck get completely dried. Okay, so any pathogen which is in the leaf and the foliage was trying to get into the onion bulbs with a dried neck without too many moisture, it will not be able to travel from the infected leaf to the onion bulb. So it's just a race of a pathogen to get into the bulbs and race of a onion bulb to tighten or dry its neck. Now, we recommend at least 3 days of field curing under sun. And for field curing, you have to clip onions. So clipping onions is also we have seen if you clip the onions at a 2 inch and leaving the 2 inch onion neck, dried neck, it will reduce the onion bulb rot internally. If you leave any length lower than 2 inches, you're increasing the risk of internal bulb rot because lower shorter neck, what happens if even if there is any kind of contamination of those neck, dried neck, there's a possibility it will get an access to the onion bulb or onion shoulder much easier compared to if you have a longer dried neck. So these steps, if you keep these steps, more likely you will have a lesser risk of developing bulb rot, internal bulb rot. Now, if the infection is coming from outside, impact, and it's also rotting your bulb from outside, those bulbs will be sorted out during grading process. Those are easier to be sorted out. But our major problem is those bulbs where the bacteria propagate without any symptoms from outside, and more likely they get entry through those onion necks. So if you have a dried onion neck at a decent length of 2 inches or more likely you're reducing your chances of getting internal bulb rot.
Fascinating. I'm taking notes for my garden since I grow onions every year and I've had a few problems. So these tips are just great to know. Dr. Isaac, do you have anything to add about the management of disease in organic onions?
Well, I agree with Dr. Dutta's assessment of the harvesting process in onions, and in Texas, that's pretty much they do the same steps as well. One other thing that's employed very commonly is crop rotation, taking it out of onions the following season and putting in some other crop.
Hmm. Okay. So I guess what are your plans for the coming season, for the coming research season?
Sure, I will start. So we are in our second year of our project. We are repeating our soil sterilization and soil biofumigation studies. We'll After this season is over, we'll discuss our results with our stakeholder advisory panel, and then we'll tease apart some of the important findings and take it to our following integration objective. So another aspect which we are studying next year is how we can introduce microbial soil biofumigation. So we have produced some of the fascinating work through this project where we have utilized some microbial biofumigation, and it has shown much better promise compared to biofumigation itself, as well as soil sterilization in reducing Burkholderia. So we will be repeating those experiments again. Now those are proprietary material. I cannot divulge those names, but these materials have shown better promise in at least in two different locations, and we're uh, repeating it. Also, we are following up or working together with our soil ecologist, food safety specialist, and our horticulturist on the impact of soil nitrogen sources and how it's impacting the soil health and so forth. So that those are my plans.
Okay, great. Dr. Isaac, do you want to talk about what you're up to for the next coming season?
Yeah, so we're in the process of doing the parallel experiments, the large scale experiments that are being done in Georgia. Right now, the second soil solarization experiment is underway to monitor survival of Burkholderia gladioli. And this week, it looks like the Overton, the East Texas experiment looking at biofumigation will get underway when they plow under the biofumigant crop, and we provide them with the infested onions to let that process start. But also, my graduate student, Jillian Hamilton, has been doing side experiments on a much smaller scale, like mini solarization experiments in little plots with different soil types uh, to look at do we see the same effects as what we see in the larger scale experiments. And one of the things that I notice is a big difference in the soil solarization experiments. What they do in Georgia or trying to do in Georgia, I think in Texas we have a little bit more latitude in subjecting fallow onion soils to higher soil temperatures, since it's possible when they harvest the onions in Texas, in South Texas, in late April or early May. They have summer months to do solarization. There's much higher soil temperatures, and they could get an effect, possibly get a better effect from that. But anyways, we are planning to do smaller experiments to validate that effect.
Sounds great. Okay, do either of you have anything else to add?
So yeah, what I would say, it has been a fascinating journey. We are learning every day something. So as I mentioned, the proposal is just an idea, but as we uh, start studying those and conducting those trials, we learn new things. And one of the major outcomes which in 1.5 years, which I've found, is these microbial biofumigants. That was fascinating. It not only reduces Burkholderia population, but also it has not harmed other population, native population of other soil microflora, and that's amazing to see why it is so targeted. So we do not understand the specifics of it. Probably through this project in the long run, we'll be understanding how the mechanistic behavior of this particular microbial biofumigant. And the good thing is that it has now been recently OMRI listed, will be available for organic growers.
Wow, well, that does sound exciting, and it sounds like it'll be great to learn more about that as you discover how it works and discover how effective it is. We'll be posting future updates about this project and conducting future podcasts, and all those will be available on our website at eorganic.info/onion. I'd like to thank you both, Dr. Dutta and Dr. Isakit, for sharing information about the project today, and thank you also to Pooja Malhotra from the University of Georgia for her help in organizing this session. We'll look forward to more information and updates about the Onion IQ project as the research progresses.